Okay, so again starting by updating the toolbox to the latest version. So latest version should be version 24.4.0b. So if you see this on the top, you have the latest version. The end of the semester, so I will spend some time to lead you go through some of the features we haven't been talking about. There are many procedures or analysis you can do I haven't mentioned, or we don't have time in previous weeks. According to our agenda today, we will start by looking at a source code for demonstration of Laplacian eigenmap. If you Google search, you can find this source code in MATLAB Central. MATLAB Central is a user discussion group, basically. If you have something you want to share as a user, you can upload your code into MATLAB Central. But you need to register as a free registration. So for your convenience, I just download this source code and put in this channel. So if you double click, open it, that's the source code. What we can do is we can just copy those source code, different lines, and then paste into MATLAB. So we look at the step by steps. This source code started by creating an example data set. It's called Swiss roll, and if you copy this line 1 to 10, and you will create this data set. We don't need a toolbox at this moment. So this data contains 2,048 points and 3 coordinates. So this is 3D data points. For example, to do a figure, if you do plot 3, you put x in, y in, z in, you should have this figure. This is not the best way to show. Okay, when I say plot, then the points will be linked by the lines. So if I say, okay, we don't need a line, I only need those markers, and then you will get this, okay? So this is a data set we just create using a simulation or the code generated. It is a simulation because there are a random function here to generate z first. Then, according to the value, then you have x, y, do this rotation, okay? So we can come back to this source code example. They are not using the plot 3. They use a different function called scatter 3. It's doing almost the same. The only difference is they gave another variable called CMap, so to give the color for each of the point. Based on the index, okay? So we can copy this and paste into the command line. So same information, it's just use different function. One is plot 3, and this is called scatter 3, also XYZ. 20 is the size of this marker. Is given by from one end to another, depends on which data has been firstly generated, put into the array. So this automatically gave you this rainbow color defined in this jet. But if you use a different color map, I can use. Remember, we have summer, right? So summer is also gave you the color map function, the coordinates. If you do this, you get a different color schema, okay? Depends on the marker, depends on the color code in that map. But the whole purpose is just to let you know some data points are from one end, some from another end. Now, if you look at the source code here, this piece of code is start with build KNN. It's a network, I believe. So we can copy from line 17 up to line 38, okay? So within this block of source code, you can find there's place where the distance between data points was computed. So that's here, okay? That's called the p distance between data points. So that's the pairwise distance between all the points. And then from there, they have a cutoff probably, and then there's the transformation. There's the Gaussian function to transform those distance into a weight. So I copied the code here, and you shouldn't see anything once you put in there, but you create a W matrix here, okay? Because we have 2048 points, so the pairwise distance is between 2048 by 2048. And then after your transformation, after you do this weight calculation, then your weight matrix is 2048 by 2048. it's a sparse matrix, so contains a lot of zeros. Then you use a sparse data type to represent. Okay, so Laplacian eigenmap, if you remember, in the middle, we need to compute Laplacian matrix that I think here they called DO. Okay, then we do this eigen decomposition, okay, eigenvalue, and then use eigenvalues as a projection. So this will create a figure of a projected. Okay, so we can copy from line 40 to 55, 54. Okay, what they showed here, because you reduce the dimension, and you have here I think this phi 2 and 3 are the two of the eigenvectors they selected, and they use 2 and 4 here, and then you have 3 and 4. So several different combinations of those eigenvectors. That's the index, okay. Then at the end, we create. So this is the code from line 56 to line 61. So let's put the two figures side by side. So it's before dimension reduction and after. So this example led you to look at the source code in there and also compare with the theory. So the theory is to build a K network because you compute the distance between points first, then transform it and take this Gaussian kernel, and then compute the Laplacian matrix. So all the information is from this distribution of data, but transformed. The relationship between data points has been learned, and build the network transformed and get the Laplacian matrix and the eigenvector problem to find eigenvectors for the Laplacian matrix, and then use the combinations of different eigenvectors to represent the position of all the points, and their relationship has been built into Laplacian matrix, and so this embedding based on that will show the relationship, but not like a PCA. We'll just learn directly where you can display the maximized variation without considering inter relationship between data points. So in this nonlinear embedding, we take advantage to learn the relationship between data points, to find the neighbors, and those information have been transferred, and at the end, you give this more realistic, nonlinear embedding results. Okay? Do you have any questions when you run this? You can copy everything and then paste here. Okay, so I'm going to clean this. Next, we are going to click this link to visit a machine learning and the dimensionality reduction, manifold learning toolbox. So Laplacian eigenmap is one of the many manifold learning algorithms. This MATLAB toolbox is designed to implement many different manifold learning algorithms. We mentioned isomap in the class, and also the number 10 here is Laplacian eigenmap. There are many others. Okay. So this is like a third-party toolbox. It's not published by MathWorks, the company produced the MATLAB. But this is like a user contribution. They developed the toolbox. They share the source code just as I did to share my toolbox. So we can learn how to download and then unzip and see what's in there. Okay. So let's click the download. Click here to download this toolbox. If you can see the link. Okay. The toolbox has been downloaded and saved in my download folder. And then if you look at the extension of this file, it's GZ and the tar. So this is a zipped format from the table, and also zipped format using a Linux system. We can still unzip it here. So we can right click and use 7-zip to extract here. Okay, the first extraction will give you the tar file. So this file will become with the extension TAR. Okay, so this is first step. So that's why it's important for you to allow this extension be shown in your system, so you know what file it is. So I extract the first time, extract here. And then on my screen, at the end, there's, I don't know if you can see over there. So this one with a GZ and become a file without a GZ. So we have to unzip this again to open it. Okay, so 2 steps. So now you click this tar file and then to do, for example, extract to this folder. Now you should have this folder in your download. So if you click go into this folder, you will see this source code and the MATLAB code and the 2 subfolders. Yeah. So we download this toolbox and also opened it. Now, copy this parse because we are going to change the working folder from MATLAB to here. So if you can select and copy this, come back to MATLAB here, and we do a CD, change directory to this folder, and I jump to this folder. So all you can change from here, and you can paste to your folder. You just copy it. So all the function under this folder become accessible for us. We can run, for example, one of the test toolbox, whatever. Okay, but the most of function are included inside this so-called techniques. If you open the technique subfolder, you will see many implementations of different functions. Okay, so this is 2 subfolders. In order to have access to these subfolders, we need to select these 2 folders and then add to set parse. We need to add 2 folders. We need to select these 2. I select the first one and then add another one. Okay, so remember the place I click is the set parse, and then you have to add 2 subfolders into your search parse here. So the function which inside these 2 subfolders will be accessible from any place. Okay? We do not want to save this in future session. We just try once, so you can click no here. We temporarily add these 2 subfolders here, but we don't need to do that again. Okay. So if you have done this successfully, these 2 subfolders will look slightly different. Before it was with a color, it's a faded color, but now you will see this full color, and that means these 2 folders, all the functions have been included in the environment. So for instance, a function inside technique, like CCA, I should be able to find it from outside here. So if I see which CCA, the MATLAB should point me to technique, this toolbox. So in that case, that indicates the parts have been added successfully. Now we can try the function this toolbox provided. The one we can start is this called DR GUI. Okay, DR means dimensionality reduction. GUI is a user interface. So author and gave our interface, a simple interface like this. Okay. So this allows us to select example data sets and using different algorithms to do the dimensionality reduction, okay? And then plot some results. You can click the first example. I hope everything works fine. It should, okay? There are 1,000 data points. So you select the first example data, and then after that, the data is loaded. And you select the second, and here you can select the dimensionality estimate, okay, estimation, whatever. A function try to estimate the number of the dimensionality is the best for you example data, and then compute mapping, and here you have many different selection algorithm to select from. So again, I will do the Laplacian eigenmap, reduce the dimension, and then scatter plot the original data. Okay, some warning message, but I don't care. So this is the data set before, and this is the data set after. Okay. I can try different data set, helix, and then plot helix. Looks like this, the original data. And then this step is optional, so I jump directly to the ISO map. But anytime if you want to look at the source code, the source code is available inside the technique, the folder. But here we just have this use the interface also provided to play with some example data sets. Okay. So on my left is example data set I load from clicking this, and I use the second called helix. So this is the data set looks like, and then on your right side is the 2D embedding for this data sets, for those data using ISO map. Okay. You can try I can try PCA. I don't know whether PCA will work in this case. Load different examples, Twin Peaks. Yeah, no, sometimes probably we need to restart the program. So I just restart it. Load the Twin Peaks and then intersect. Okay, so this is just a small interface. I just play with different data sets, try different algorithms. Again, if you want to learn the details, you have to look at the source code inside the technique subfolder, and also you need to understand each steps, okay, based on the theory, if you have to study any of them. So that just look at isomap I mentioned. This would be very simple because isomap is using geodesic distance to replace Euclidean distance. Then you run MDS on it. That's isomap, right? Open. Yeah, I have trouble even to open this file. Okay, I will restart my MATLAB. Yeah, what I want to do was just like let you to open one of the implementation of the isomap algorithm, and then look at inside, see which part is most important. So here it starts by creating a nearest neighbor net graph, okay? Find nearest neighbors and search nearest neighbors for each data points, then try to create this map network first, and then start to compute the shortest distance between any given points in the nearest neighbor graph, because that's the real distance you have to move through the network from one node to another. So that's the place you compute the D matrix, the distance matrix. And then you have this basically MDS, you compute a gram matrix. If you remember MDS, this M is a gram matrix, and then you do the eigen vector problem and to find the coordinates. So this implementation directly follow the theory, and if you put your theory, the paper published in Science year 2000, and side by side you will see, okay, everything makes sense. All right, we are going to finish off this part. We need to switch back to CDGEA, switch back the working folder back to toolbox. So you type this function will lead you back to there. Okay. And also I want you to make a which PCA. So I just want to make sure the function PCA is pointed to MATLAB toolbox in statistic toolbox, rather than the one we just download, because we just download the toolbox contain a function called PCA. So the same name, if you have that in the parse, then you will confuse MATLAB, say which PCA function you want to call. So if you type which PCA here, and you get this correct version in MATLAB provided built-in. This is correct. Otherwise, you can see I shut down this and restart my machine, not a machine, the MATLAB. Yeah. Or you can, in the set parse, just remove the DR toolbox, the two parts we just add. You can remove them. Then you should have the correct parts here. Okay. So let's move to another function called magic, because remember magic using this manifold learning technique to do imputation. We will play with this algorithm and see what it does to your data. All right? So in this case, we need example data. We only need a big X. This function doesn't care what the name of genes. To the function, it's just a matrix with 0, 1s, and the UMIs, a lot of zeros. It will do the imputation. Okay, rows are the genes, columns are the cells. So we just need a big X, so we can just get it from the example. So you can load this data, example data, and export. Export data to the workspace. Then you select I need X and G. So X is the expression matrix. G contains the name of genes, although we don't need it, but just export to the workspace. Okay, so now this X is here. We have this number of genes, this number of cells, so your X is here. This X is sparse matrix, so you don't see them, the values, but you can change by using a function called fold, and this will convert this sparse matrix in a full matrix to display all the values, even though many of them are zeros. Okay. Now we don't need this interface. You can shut this down because we already export and save the variables we need, X and G, in the workspace. So remember, we have this matrix. For instance, we want to compute co-expression or correlation coefficient between two genes. Can anyone tell me how can get the name of the first gene? You can click, so the first gene name is XKR4, okay? Or you can see, okay, the G is my list of genes, the first one, first item in there. So I can get the name of the first gene. And I can get the second gene's name by this, okay? The second gene. And you also know how to extract the gene expression from the matrix X. So the first gene expression will be the first row. Okay, so this is called X1. So this function extracts the first row that this XKR4 genes expression across 8,000 cells. You can do this for the second gene, okay, gene 2. Now we have two genes expression profile. We want to see whether in a cell you have first gene expression, then the second gene also expressed, or whether they correlated. Normally, we can do a scatter plot. So scatter, put X1 and X2 here, okay? But you will see this is pretty empty because many cells have zero expression. Yeah, you don't know how many cells has this 1 expression, how many have 2 0, how many have. So you see the data points, but there's no way you can establish any correlation between these two genes expression. This is true when you apply to any pairs of genes in mostly, because it's so sparse, right? We can find some genes maybe highly expressed just by work through this matrix. Let's see. Okay, my gene number 206 looks highly expressed. Okay, so please find out which gene, what's the name of this gene, and make a variable okay, extract this gene's expression, we are going to plot this scatter plot with another highly expressed gene. So let's see, my first X1 now becomes not the first gene, but this 206, okay? So what's the name of this gene? We have to check. Name of 206. This is, okay, EF1B2. So I can move to another highly expressed gene. Let's pick this number 276. So this gene, 76, is RESP18. Okay, let's call it X2. So it's not the second gene anymore, it's 206. All right, so I have my manually picked two genes, and by looking at this table, and these two genes looks highly expressed across the cells. So I can extract their expression and look at their scatter plot. Okay, so this picture looks very different from the one that we randomly selected two. And the gene 1 and 2 was lowly expressed. There are too many zeros. We don't see any correlation, but these two we manually selected. They are highly expressed genes, so you start to see some of these patterns. There are some cells have one gene is 22, another gene is 358. So you have this scatter plot. So we can do this for any given pairs of genes. See which two are positively correlated. These two genes looks positively correlated to me. But the correlation, if you insert a basic fitting, you can fit a line here. Yeah, we know this slope, okay, is a positive 2.339. So this is a linear regression, try to build a line and a regression line to fit all the data. But because of the sparsity, and we don't really have very good estimation of this. Okay, so here this comes into the problem of imputation. So we can use manifold learning to smooth the relationship between cells and using the information between neighbor cells to fill in many of those zeros. Yeah, question, Sarah? You can. There's a function, CORR, correlation. So if you put, you can compute this pairwise correlation coefficient as a whole. Then you pick which two are the most correlated. Yeah. Yeah. Their function you should be able to use, compute, not manually pick, but compute all pairwise correlation to help you. Again, because this sparsity issue, so you can do this pairwise correlation, but oftentimes it doesn't make strong sense. So that's why imputation may help. So we are going to do the imputation for the matrix. Okay, so you gave X to the algorithm. The algorithm will return you an imputed version of X. So now we call it X imputed. Let's call it XM. Okay, imputed. So all you need to do is to run point empty. Empty means MATLAB. Okay, underscore magic. Capital. This only takes one input variable. So you gave X, that algorithm gave you impure version. Okay, now in your workspace, you have another X, it's called XM, have the same size, same dimension with original X. So this is our original X here. If you double click this, you get this imputed. Okay, if I plot say, a block of the, let me close all the figures. If you use ImageSC to plot X, you select, for example, 1 to 200, 1 to 300. So here I just extract a small corner of the X matrix and look at its expression. So this is before imputation, okay? Many, many zeros. If you do this for the XM imputed version, you have color bar. So you have this smooth version of those the matrix. Okay. Now we can do this for the two genes we selected. The first one is this, and the second, not using the original version, but use this imputed version. The same, but I just use imputed version to get the two genes expression. Okay. Before it's big X, this original version before imputation, but then now I change it to XM. Then you can run the scatter plot between these two genes. Okay, you show this, it shows this. Okay, so there's the imputed results to plot these two highly expressed genes. You can try this for gene 1 and 2, okay, before you cannot figure out anything between gene 1 and 4, gene 1 and 2. Let's do this. First one, gene 1, use imputed version. X2, number 2 genes, and then plot that. Okay. So this is very different from the previous before imputation. So this imputation procedures changed drastically. Okay, some hidden relationship between genes may be discovered. Okay, so for example, within these two genes, this is the two highly variable genes we selected. Probably there's a subcluster of cells in this group here, does not show the correlation between these two genes. But the other type of cells in that group, you do see this. Okay. So this is to help you to identify those nonlinear relationship sometimes. If you can plot like 3 genes relationship after imputation, you see this change of relationship. Sometimes it's positive, sometimes negative, and depends on which subcluster of cells, maybe conditioned on the third gene's expression. You see those nonlinear relationships. This is through the help of the imputation. If you do this on the unimputated data, you don't see those relationship at all. Okay, let's take 5 minutes break. So imputation in some special circumstances, and you can use it to enhance the signal. The problem is we don't know what's the impact of the imputation to the real signal of the biology. Okay, so people use this with some caution, and you don't want to try to purely trust the imputed version of the data, because it does give the drastic change of the relationship between genes, between cells. So in the toolbox, I don't have, I can certainly set up a button to allow you to do imputation to replace this sparse matrix. But I decided not to do so because the reason. We do have some function in there that we can select in the middle of some of the operation. You can decide you choose to show the imputed version. For instance, in this here, you can click this violin button, and here you see I want to look at some genes expression across different cell type. This is a grouping variable that will be in your x-axis later. So you select this, and you see I want to look at some genes expression. So you select expression, and then paste a list of genes. Here it is a random set of genes. Then you click OK. Now you have the option to see whether you want to do the imputation before you show this violin plot. If not, let's see. If not, I will. Let's see yes. And in the very bottom here, you have a magic function to do the imputation. So your value will be changed. The imputed value will be shown in the violin plot at the end. So if you look at these steps, behind the scenes, this magic function has been called, and then the X matrix becomes XM matrix, and then the expression was computed. So that's the result of the violin plot. On your y-axis are the different cell type, because we select the cell type as the grouping variable. So here, cells have been separated by their cell type. And the y-axis are this particular gene's expression value in the cell, but the value has been imputed. That's why you see this nice violin shape because the imputed value gave you some of this continuous feeling, and otherwise you have a lot of those genes have zeros, then your violin will looks not like a violin. It's like well, okay, yeah. So for the visualization purpose only, okay. Yes, so the y-axis are the expression level. There are many cells in this group. So I show you an example. Each violin here contains maybe several hundred of cells. And the wider the place, are the place where most of the cells have that value of expression for this gene. And the narrower, the tips are some tips created by a few cells have that particular expression, and they look like outliers. And this distribution, for example, the purple one in the endothelium cell is very different from this neural cells, because neural cells, most of the cells has expression level 0.2 after imputation. The shape is different from the standard violin. You have a mean in the middle, and then most of the cell have a distribution around that region. But this distribution is very different. It's enriched in the top layer of this region. So violin plot gave you the shape to give you the idea where the cells distributed. Okay, so I'm going to do this without imputation and to let you know what it looks like if you do the raw data. Expression, paste some genes, like example again, but as a representative. Here I see no normalization, no transform, no imputation. Then the violin plot you'll get is from the UMI count. Okay, many of these genes have 80%, 70% of zeros. That's what you get because most of them are zeros. So this is very, very useless because you cannot compare any meaningful values across subgroups. All right. So in this case, for the demonstration, for the illustration purpose, you want to see, okay, some of the genes express really high in particular given cell type, and then you know this is true. And you can see, okay, I use imputated version rather than the raw UMI count to do the violin plot. Questions? Yes? Yeah, so this is a gene expression. You can do this violin plot for many genes. So this is selected. Mm-hmm. Whole cell expression. You mean one group to another compare? You should be able to. Yeah, so we are talking about the differential expression. So in the toolbox, you click this and to compare two groups. For example, you compare between two cell types and then select these two B cell against this, and you can have cross two groups to compare. But when we do the comparison, we don't do the imputation. But you can do the imputation then compare if you trust the imputation results. No, but this can help you to select which gene you want to plot a value in. For instance, here I compare B cell with macrophage, so I export this. So I know which one is mostly differentially expressed, so I can select those genes, this table here. So I can select those top ones. Remember the name and then come back to here to do the violin plot. Cell type, violin between the gene, I just do the DE, I can do imputation. And I expect to see the B cell and the macrophage have a very different expression for those selected because I run the DE analysis as a group. So the difference is between macrophage and the B cell for CCR10 macrophage D cell. So that's what we expect to see. Okay, so we move to tensors. Okay, let's clean this, close. We know how to create zeros. That's a 2 by 3, for example. We know how to create a matrix. I just generate a matrix with 2 rows and 3 columns. I can fill in the values. See, okay, 2, 3, this position, I gave you a value number 5. All right? So you know this. You know how to do SVD and PCA on those matrices. You can also generate let's see, 4, 3, 2, okay? You can also generate higher dimension tensors in MATLAB. You just put more level of this dimensionality. You can even have a higher, okay, another layer you can, but I just use 3D. So now you create a tensor with 4 by 3 by 2. To show this, if you double click, you get the fourth layer of the third dimension. So you have a first matrix 4 by 3, layer 1, and layer 2. Okay? So you can put values in here. So I can put 2, 3, 1, okay, this position as 5. This is a display, and this is a tensor I created with a big X. I have 2 layers. Each layer is a 4 by 3 matrix. And then in the position of the first layer and the position 3 and 2, I put a value of 5. The rest of them are all zeros. Yeah, it's clear on your mind. You have this picture. So this data set, this representation is a tensor. We can create a random. If you do this, it will not fill zeros, or you can give ones. Okay, those functions, they all available for tensor purpose. Okay, so in this example, you use the function ones to create this tensor fields with ones, and then you can use random. This gave you a set of random numbers between 0 and 1. Is that okay? Okay. Now let's come back to another example. I will create a random 25 by 25. So this is a matrix, right? So this matrix I can plot and to see the values using a function called image as C, okay? So if I put this X in here, I will have this picture. It's 25 by 25. All right, so you know how to do this. And the values in there, it will be indicated by the color. And the blue ones are those close to 0. And on the top, those yellow ones are close to 1. Now, I'm going to give you a tensor rather than now x becomes a tensor. You can think about this as a 25 by 25 picture, this block picture, but you have 9 layers. So you cannot use this to run it because this function require a 2 by 2, require a matrix. Okay, so we are going to do a loop. I will extract the first layer and plot, and then move to second layer and plot all these 9 different pictures out. So in this case, I can extract the first layer by using this, and the first one. So this should work. This will extract from the tensor, there's 9 different layers. But this will extract the first layer and to plot something like this. And I can do the same thing for the second layer by giving 2. Okay, they look similar, but the values are different. Okay, the random values. So I'm going to do a loop through all the from 1 to 9 and put all the 9 pictures side by side. So I'm going to create a loop. So for k goes from 1 to 9. Okay. And then for the figure, there's a function called next tile. And then I start to put image to replace this into a variable k, okay? So through the loop from 1 to 9, each time we loop through and at the end. So I close the loop with the for and end. So these 4 lines create a block. This block will create this picture. So image SC cannot plot more than 2D, but I just separate those multiple layers into 9 different subplots, and they're showing on this picture here. Now I'm going to do some of the operation on the matrix itself. The operation I'm doing is something like this. So I will replace from my different layers for each of these x. But right now x is purely random, but if I select 1 to 10 and 1 to 10 this layer and gave a value k. Okay, I changed each layers of the one corner of this matrix. It's from corner 110, 110. So this corner on your upper left has been replaced. All the random value over there has been replaced with the layer index. So the first layer I will give 1, and then move to another, I gave 2. Okay, so now I changed the values in the tensor, and then I can plot that again. Let's close all first and run this block again, so I can copy this plot. Get some values here. Let's see. I need to check my So this is my first layer of data. It's all nice. Second, yeah, I think this should be K. Yeah, so that's what. Yeah, so I replace this. The purpose is I try to get a different corner, have different values. But I had a typo. So if I replace this with a K, this should create the data I really want, the structure, I hope. Yeah, so if you want to start from scratch, I show you, if you want to start from random here, okay, so this is the first line, and this is the second, third rows here, okay? And then you can have the plot, the close all. Next tile, image. Yeah, I want to create some gradients, the change, okay? Using this. So if you want to start from scratch, you can just regenerate those random tensor and then change this corner, the values. The first one will be 1, the second one will be 2, and here will be 9 filled in using this code here. And then just plot it out. So I created this because I want to show. So for this data set, this 3D, your x, y direction has no relationship among data points. They're all random. But if you look at the z axis, which is between layers from 1 to 9, there's trend in this corner here. This is 1, and this is 2. The values in there changed accordingly, okay? Because the value changed, so you see the contrast between this value with the rest of random are different. So if I put the color bar in each of this, you will see the highest value is changed from same as the index. You can pay attention to the scales. So this is the value in this matrix is from 0 to 1, but on the top. And for this matrix, the top value is 2, okay, 0 to 2. This is from 0 to 3, the 4, 5, until 9, okay? Okay, so most of the function we have like eigen or SVD, which you apply to matrix. As I said, SVD you can apply to any matrix, but here, because we are dealing with tensor, so you cannot do SVD directly to learn those multi-dimensional components. All right. So the native support for tensor operation, there's not many functions you can have from MATLAB itself. So you need to rely on the third-party so-called tensor toolbox, MATLAB. Okay. In this case, we don't have to download it because I already put this toolbox in single cell GA toolbox. So if you look at the folder inside here, there's tensor toolbox. So what you can do is just browse down and double click this. You should have all the tensor related function in there. Now we need to convert this X, the 3D matrix, into a format which the tensor toolbox can work on that. So we need to do a tensor function, give the X, so convert this 3D matrix to a tensor that's called XT, okay? So that's because we have the support functions from this toolbox. We have XT, which is object, has been built as object. So all the functions in this toolbox can apply to this XT. Just like in SCE, I defined in my toolbox as a single cell experiment, or that's object, then a lot of function can handle that. So now we have XT, we can use this alternating the CP decomposition. So what we can do to decompose this is change this to M components by using CP ALS, put XT here, and then see, okay, 3 components. Something like that. It should work, yeah. The toolbox? Oh, the toolbox is built in my toolbox. I already download and put it in there. So all you have to do is to switch yourself into this, click into. Yeah, it's already there. So you don't have to download. You double click and you are in there. Yeah, so there are a lot of information about the functions in there, and there are different type of tensor, and most commonly used the CP decomposition. We can take a look at the first function, the alternating least square algorithm. There are some others, but this one is the one you can easily use. There's examples how to use these functions. Okay, so it decomposes into M. There are different ways to visualize this. I don't know, I can try. So it's called M1. So I just rename this. I don't know what we can get. Yeah, so the toolbox creates some this component view, and you can start to see we do have a separation in that particular corner over there, and then these components have been learned. The 1 to 10, there's some different from the rest of the matrix, and then there's gradients you can see from 0 from the first, second to the ninth layer. So those information have been learned and decomposed into different components and their values. Okay, so what I also do is to do this denoised version. So because you have all the components already learned from the CP decomposition for the tensor, and we can use that information to reconstruct your the matrix. Let's see, reconstruct M. So let's see X. Yeah, so our original is X 25 by 25. So this will be imputed. So we use this tensor learned components to reconstruct and get the XM. Then we can plot out the figures, okay? Let me end this. Let's close all. And the next tile, image. But this time I'm using XM. So this is after decomposition and reconstruct. Let's see what the figure looks like. So compared to the original, you see this is much more smooth. There's a lot of random noise has been smoothed out. But the major pattern, which is increase of the values from layer 1 to 9, has been kept. So this is a smoothed version, denoised version of my original data with a lot of randomness. So that's the use of this function to do the tensor decomposition and denoise. So you see this much clearer signal. Yeah, so this is a very quick introduction of the very basic use of the function. So using this constructed examples. So if you, in future, you study, you have those multi-layers of the information, and you know in some direction you should learn some kind of patterns. Right? think about this method. Questions before we move to the final So what do you see here? The reconstruction of the full data set using components. Those are some details and how the function has been called, how to do this. But you have to have this big picture on your mind. You can do this in the similar way to do the SVD and then truncated SVD to denoise the signal. Okay, so we'll keep moving to the next section. So now we can close all C can change back to the working folder of gene expression analysis 2. The function on my schedule I want to show you is this external function called geometric sketching. This is a subsampling program, and this is in Python, and if you set up your environment correctly, you should be able to use this directly. It will subsample, for example, we have 8,000 cells, but it will subsample about 4,000 cells. Why you are using this? Because when you have large data set, it slows down everything. You want to have a quick impression of what cells in there, what's a quick relationship between that before you end up with the final results. So in order to facilitate those quick analysis, you can do a subsampling. I don't need 8,000 cells to tell me the major signal. I only need 4,000 or even less. I can do this again to select 2,000 cells. The easiest one is to randomly select, okay, 0, 0, 1s, and to select randomly. The argument about this algorithm is if you randomly select, then some population of cells like this corner here, if they're real cell type, then you probably will never get enough cells for representative signal to give some of those real situation. So your purely random selection will get rid of a lot of those outlier cell types. But this geometric sketching approach try to give different weight. They will sample down most from the large population, but they keep some of those representative small population, small subclusters. But the cost of this more sophisticated sampling is much slower. And for purely random selection, it's taken no time. You just randomly select half of the cells. But this will take some time. But I just show you how it runs on my computer. I haven't been running this before, so the program probably have to find where my Python is installed and install this package. So that may take some time. And the PubMed ID is associated with this the article. You can find the details in how this geometric subsampling is designed or implemented. I start to hear the noise of some of the computers. It's not If you want to use random selection, you can have in the edit the menu. I believe there's items allow you to select to subsample the cells to 50%. Then you can select whether it is randomly subsampling or using this method. If you do this 50% subsampling twice, then you end up with only 1/4 of the original data set. Sometimes it's essential for you to run on your local machine and to quick discover the patterns. You can always come back and say, I know those patterns I want to publish, and you use the full data set to run on the clusters or other computers with large memories to finalize your results. But to quick learn the biology, you probably want to use the subsampling data. Especially today, we get thousands, sometimes millions of data. My computer can easily handle close to 1 million cells. I tried, but each step is too slow. You want to subsample cells. Yeah, I think on my machine, this package is just installed. Now, the cells have been, numbers have been reduced to 4,000 something, okay? Yeah, so that's about it. So after throw away half numbers of cells, you want to run this QC again to get rid of some genes if they're not highly expressed enough. Okay, we run the QC again to remove 329 genes in my example here. So next week, I will be here a little bit late. So Chakata and Luis will lead you to work on this network analysis. We do have some buttons here, and you see this is for network building. Next Thursday. Tuesday will be Thursday. I will be here a little bit late. So Chakata and Luis will help me to set up and start the tutorial on time. Yes, we do have some network-related function. We will talk about those networks and what you can do with networks next week, Tuesday. Okay, yeah. So I think that's most of the things I want to cover. So your homework is about the DV analysis, and we also have the differential program analysis, which consider gene set difference between different group of cells. You can have two conditions, then compare not only differential expression, individual gene based, it's on the whole set of genes and compare. You can run those across different cell types. So if you focus on B cell first between two groups, move to T cell to alpha cell. So depends on how many subtypes of the cell types you want to run them continuously one after another. So there's a shortcut you can run in the batch mode and to iterate through all the cell types in there. Otherwise, you can click or extract one cell type, do the DE first, another cell type, do the DE. You will achieve the same results. Okay, so I think that's most analysis we will have. As I mentioned, there are subsample procedures on here if you want to subsample cells to 50%. And you can select uniform subsampling or the method I just showed you. So when you click this uniform subsampling, this 4,000 cells will become 2,000 something just by one click, okay? Because this is so simple operation. All right. See you next Tuesday, and have a good weekend. Any questions? Quick questions? Okay. Yeah. Which one? Which two? HG. Which two? The two box? This one? The two box? Yeah, I always, you download it, you use it every week. It is open sourced, and you can make a change you like.